In this video, we will discuss about some of these AMT applications on the pancreatic and breast cancer cells. So AMT means this adipose manipulation transplantation. It can cause tumor suppressions in the pancreatic cancer and the breast cancer. And we have discussed about this AMT in our previous videos. You can refer to those videos to understand what does this adipose manipulation transplantation means. In brief, they will inject these genes that will cause high nutrients consumptions by this adipose and thereby leave the nearby cancer cells deprived of the nutrients that will cause the suppressions of the tumor growth. So what it is, in the case of pancreatic cancer cells, they took a mice model that has these KRAS mutations and P53 mutations, and they use this PDX CRE ER based model where the these mutations are triggered with this reagent. The name of the reagent is in the paper. And so 6 weeks after these injections and triggering of this pancreatic cancer, what they did is they check the tumor weight, growth, metabolic genes, hypoxia, angiogenesis. And one thing that I forgot to tell you, that once they triggered this pancreatic cancer mutations, they inject this adipose, which can upregulate this gene UCP1 genes. And 6 weeks after this these adipose injections, they check these parameters, that is tumor weight, growth, metabolic genes, hypoxia, angiogenesis. And in terms of the tumor weight, they are able to see that tumors that are co-transplanted in this adipose, which has this UCP1 genes upregulated, cause low tumor growth compared to the control, where they use VP64 genes that is upregulated, which does not cause high nutrient consumptions by the adipose. And in the previous video, I have misspelled VP64 as VP94. So it's actually this VP64 that has been transplanted in the control adipose, and in the case of this actual experiment, they transplant this UCP1 gene in the adipose. Now come back to the topic. In the panel C, what they check is they check the tumor mass, and the tumor mass is significantly lower in the case of this adipose with this UCP1 gene upregulated, and the CK19 signals are also lower for this actual case, that where adipose is transduced with this UCP1 genes, or where UCP1 gene is upregulated in the adipose. And the CK19 is one of the cancerous markers. When they check the proliferation markers, that is MI67, GLUT2, CD36, and CPT1B, they are able to see they are also down-regulated in the case of tumors which is co-transplanted with this adipose in which UCP1 gene is upregulated. But for the GCK, they do not see any significant changes. So this is quite surprising, we can say that. And when they check another proliferation markers, that is Ki67, and this also goes down in the case of this tumors co-transplanted with this adipose in which UCP1 gene is upregulated. And the hypoxia marker, that is CA9, and this angiogenesis marker CD31, these are also goes down as expected, because these are also related to the tumor progressions. So for this pancreatic cancer cells, they see the expected result, except for this GCK1, that when the adipose with high nutrient consumptions are transfected near the pancreatic tumor cells, it will suppress the pancreatic tumor's growth. And the similar experiments in the female mice model, where they first inject this breast cancer, that is MMTV, PYMT, and then they plant this adipose at two different sites. First one is this dorsal site, another one is this mammary gland site. It can be seen here. And then they check after 6 weeks tumor volume, growth, metabolic genes, hypoxia, angiogenesis. And surprisingly, in both the cases, what they are able to see, the tumor growth goes down. So for the breast cancers, it works for at both the cases. It works when the adipose is transplanted at both the locations. And when they check the tumor volumes, it also goes down in both the cases. And similarly, the proliferation markers, that is MKI67, the GLUT4, GCK, CD36, CPT1B also goes down for the case where breast cells are co-transplanted with the adipose in which UCP1 is upregulated. And these are the immunofluorescence image of the Ki67. You can see that in the co-transplanted with this adipose in which UCP1 is upregulated, we can see a decrease in the Ki67 signals in both mammary gland transfections and dorsal transfections. The CA19 signals, that is hypoxia signals, also goes down compared to its control. The CD31, which is for the angiogenesis, this also goes down, and the apoptotic factors goes up for both the cases, indicating that it's the apoptosis that get upregulated and the tumor progressions get suppressed. And for the pancreatic cancer also, the apoptotic factors goes up, that is caspase 3 goes up, and those results are in the supplemental figures. And the quantifications of these immunofluorescence images in the panel J, K, LM are in the bottom side of the their respective immunofluorescence. That is for the case of mammary gland and dorsal transfections, we can see that Ki67 is statistically lower, and similarly for the CA9 plus, CD33, CD31 plus, and our signals are also lower. And for the caspase 3, we can see the signals are higher, where the adipose with UDP1 upregulated genes are co-transplanted with the tumors. Both of these studies shows that their AMT transplantations can be used as a therapeutic approach to suppress the tumor progressions. So I hope you understood their results. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.